The synthesis of biological polymers requires an energy input. The macromolecules of the cell constitute the vast majority of its dry mass, that is, the mass not due to water. These molecules are made from subunits or monomers that are linked together by bonds formed during an enzyme catalyzed condensation reaction. The reverse reaction, the breakdown of polymers, occurs through enzyme catalyzed hydrolysis. This hydrolysis reaction is energetically favorable, whereas the biosynthetic reactions require an energy input and are more complex. Figure 338. The nucleic acids, DNA and RNA, proteins, and polysaccharides are all polymers that are produced by the repeated addition of a subunit onto one end of a growing chain. The mode of synthesis of each of these macromolecules is outlined in Figure 339. As indicated, the condensation step in each case depends on energy from the hydrolysis of a nucleoside triphosphate, and yet, except for the nucleic acids, there are no phosphate groups left in the final product molecules. How then are the reactions that release the energy of ATP hydrolysis coupled to polymer synthesis? For each type of macromolecule, an enzyme-catalyzed pathway exists which resembles that discussed previously for the synthesis of the amino acid glutamine. See Figure 333. The principle is exactly the same in that the OH group that will be removed in the condensation reaction is first activated by forming a high-energy linkage to a second molecule. The mechanisms used to link ATP hydrolysis to the synthesis of proteins and polysaccharides, however, are more complex than that used for glutamine synthesis. In the biosynthetic pathways leading to these macromolecules, a series of high-energy intermediates generates the final high-energy bond that is broken during the condensation step, as discussed in Chapter 7 for protein synthesis. There are limits to what each activated carrier can do in driving biosynthesis. For example, the delta G for the hydrolysis of ATP to ADP and inorganic phosphate, P subscript I, depends on the concentrations of all the reactants, and under the usual conditions in a cell, it is between -11 and -13 kcal per mole. In principle, this hydrolysis reaction can be used to drive an unfavorable reaction with a delta G of perhaps plus 10 kcal per mole, provided that a suitable reaction path is available. For some biosynthetic reactions, however, even -13 kcal per mole may be insufficient. In these cases, the path of ATP hydrolysis can be altered so that it initially produces AMP and pyrophosphate, PP subscript I, which is itself then hydrolyzed in solution in a subsequent step. Figure 340. The whole process makes available a total delta G of about -26 kcal per mole. An important biosynthetic reaction that is driven in this way, nucleic acid polynucleotide synthesis, is illustrated in Figure 341. Essential concepts. Living organisms are able to exist because of a continual input of energy. Part of this energy is used to carry out essential functions, reactions that support cellular metabolism, growth, and reproduction, and the remainder is lost in the form of heat. The primary source of energy for most living organisms is the sun. Plants, algae, and photosynthetic bacteria use solar energy to produce organic molecules from carbon dioxide. Animals obtain food by eating plants or by eating animals that feed on plants. Each of the many hundreds of chemical reactions that occur in a cell is specifically catalyzed by an enzyme. Large numbers of different enzymes work in sequence to form chains of reactions, called metabolic pathways, each performing a different function in the cell. Catabolic reactions break down food molecules through oxidative pathways and release energy. Anabolic reactions generate the many complex molecules needed by the cell, and they require an energy input. In animal cells, both the building blocks and the energy required for the anabolic reactions are obtained by catabolism. Enzymes catalyze reactions by binding to particular substrate molecules in a way that lowers the activation energy required for making and breaking specific covalent bonds. The rate at which an enzyme catalyzes a reaction depends on how rapidly it finds its substrates and how quickly the product forms and then diffuses away. These rates vary widely from one enzyme to another, and they can be measured after mixing purified enzymes and substrates together under a set of defined conditions. The only chemical reactions possible are those that increase the total amount of disorder in the universe. The free energy change for a reaction, delta G, measures this disorder, and it must be less than zero for a reaction to proceed spontaneously. The free energy change for a chemical reaction, delta G, depends on the concentrations of the reacting molecules, and it may be calculated from these concentrations if the equilibrium constant K of the reaction, or the standard free energy change, delta G degrees for the reactants, is known. Equilibrium constants govern all of the associations and dissociations that occur between macromolecules and small molecules in the cell. The larger the binding energy between two molecules, the larger the equilibrium constant, and the more likely that these molecules will be found bound to each other. By creating a reaction pathway that couples an energetically favorable reaction to an energetically unfavorable one, enzymes can make otherwise impossible chemical transformations occur. A small set of activated carrier molecules, particularly ATP, NADH, and NADPH, play a central part in these coupling events. ATP carries high-energy phosphate groups, whereas NADH and NADPH carry high-energy electrons. Food molecules provide the carbon skeletons for the formation of larger molecules. The covalent bonds of these larger molecules are typically produced in reactions that are coupled to energetically favorable bond changes in activated carrier molecules, such as ATP and NADPH.